Okay, good. Germline cells give rise to gametes. Anybody ever read Huxley's Brave New World? Okay. What was the name of the drug? Soma. Okay.
I think you put that in the wrong place.
What?
You put somatic, where it should say germline.
Somatic cells
Somatic do not give rise.
do not give rise.
Yeah,
Yeah.
because cells do give rise.
Oh, cells that do give rise. Oh, sorry. Oh, hold on a second. You're right. You're right. Here, hold on. And which ones are these? That do give rise?
Germline.
Germline. Okay. Thanks. Oh, germane. No, that's not what it is. Well, it is germ. Okay. Okay, good. So normally, we've got a normal cell. It divides. All is good. So as we were listening to that like karaoke song, cell division is great, except when it's not, like many things. Like eating ice cream is great, except when it's not. When is it not good to eat ice cream? Is there any time? No, crazy, no way. It's always good to eat ice cream. Okay. If, in fact, you might end up with cells that divide that's unregulated, forms a tumor. Not cancer, just forms a tumor. This can go one of two ways. It still is a tumor, but on the other route, it actually can invade adjacent cells. And then what's that called?
Contact inhibition.
Okay, so it loses contact inhibition, exactly, and you end up with a malignant cell. So that's the difference. So there's benign, right? Malignant, invasive, and it loses contact inhibition. It doesn't travel, non-metastatic. So it stays in place. It's probably something, it might kill you, but it's less likely to kill you than, let's say, a metastatic cancer like melanoma. And then metastatic. Means it's able, so what is it lost? To become metastatic? To travel? Anchorage dependence. Good. So, cancer cells differ from normal cells. They divide when they shouldn't. So, what is it? On the bottom, we see anchorage dependence, cell cycle control, or contact inhibition. Which one is it? Cell cycle control. Good. Okay. Invading surrounding tissues. What is that? Okay. And then finally, anchorage dependence, when it's able to metastasize and move to other parts of the body. Okay, so here we go. Radiation therapy is least effective against tumors that have lost what? Huh? C? Okay, exactly. In other words, they're usually tumor suppressor because of tumor suppressor genes. And instead of a cell having to commit suicide, the suppressor gene isn't working, and so it doesn't. So the only radiation therapy, what it does is it creates mutations, and those, normally the immune system, the tumor suppressor genes, would actually make that cell commit suicide. Okay. Why is it good to take multiple biopsies from a single tumor? C?
There's two C's.
Effective against only one part of the tumor, because unregulated cell division leads to increased. So is it just C or is it D? All of these. See, if cancer cells didn't evolve, then there wouldn't be a problem. But everything evolves. Oh, you're already doing it. Okay. So I just want to look at cell cycle stuff. If we start down at G phase, now remember, most of the time, cells spend most of their lives in G phase. Some cells spend all their time and all their life in G phase. Like, what kind of cells do that? What cells usually don't divide during your lifetime? Neurons. Okay. A lot of cells last actually for, let's say, months or years, and then they still might divide. And there's actually, it turns out, there is some division in some neuron cells. In fact, that's what they're working on in terms of spinal cord injuries, is trying to get those cells, using stem cells, to continue to divide, to reconnect. Okay? Now, that's a normal situation. When they do go on to divide, they need 2 copies of each chromosome. So remember, an individual who's diploid has a copy from their mother and a copy from their father. During synthesis, you get a double copy. You take the 1 copy from the mother and duplicate it, so now you have 2. And you take the 1 copy from the father and duplicate it, and now you have 2 of those. So you have twice as much DNA per cell as you need. That's called synthesis phase, S phase. The growth phase. To actually go through mitosis and create another cell, you can't do it out of thin air. You need resources. And the cell needs to grow and develop. And certain genes need to be either ramped up or tamped down. So it goes through this G2 phase. And remember, there's going to be cell cycle points that actually stop and re-examine cells to make sure that it's actually not going crazy. Okay? And then in mitosis, of course, we have the prophase, where the spindle fibers actually develop on either side of the cell. The nuclear envelope disintegrates, and the chromosomes, which are normally spread out, coalesce and condense into what we normally see as chromosomes. That's the only time we actually ever see them, when they're actually condensed. And then they start lining along the metaphase plate. The spindle fibers from both poles stretch out and connect to the centromeres. They're pulled apart. You're into anaphase. And then telophase is when the nuclear envelopes of the 2 new cells reform, and then you undergo cytokinesis. And then you've got two daughter cells that are now in the G1 phase. In fact, why don't we... Okay. So, in terms of mitosis, so we've got G1, S, G2, and then we've got mitosis, right? Okay. Mitosis starts out like this. Hold on. So here's mitosis. We've got this cell. We've got the different, double the amount of DNA. And in other words, we have got chromatids. So we've got all these like that. There's another one like that. Let's say from mothers and okay. So that's prophase. Okay. Then we have on either side here, and we've got these chromosomes that then line up along the metaphase plate. Okay, and these each go to the centromeres in the center. Okay, and then finally, we've got oh, sorry, I don't want to do that. No, cancel. Okay, and then let's go on to the next one. So the last one was anaphase, and now this is telophase, where we've got stop that. And this start to form, and same thing over here, start to form the nucleus, and then finally cytokinesis, where they divide into two. Yeah.
Symbiotext, that whole, the mitosis one, they did prophase, prometaphase, metaphase, but you haven't talked about prometaphase. Sure.
Oh,
It was on,
I just call it, I usually just go through.
from one to.
I just
like,
go
test-wise, it's
prophase,
metaphase.
and then metaphase. But there's actually early prophase and later prophase, where things just start to line up. So in the beginning of prophase, you actually, there's condensation, and you actually start to see the poles. And then they really start to condense, and then they start to migrate to the metaphase plate. And the spindle fibers start to connect. Okay? So that's how it works. Okay, we've already done this. Sorry. I don't know why. Oh, okay. Okay, so just so the vocabulary again. We have somatic cells and they're diploid. And they're homologous. That means you've got one chromosome, there's a total of 22 chromosomes, and then another pair of chromosomes that are not homologous, there's an X and a Y, they're germline cells, right? Okay, so homologous chromosomes are one from each parent, and they have the same genes, they might have different alleles. Okay, here's the centromeres. Centromeres. These are different genes, right? They might have different alleles. This is a non-homologous pair of chromosomes. In other words, this one might be chromosome number 3, and this might be chromosome number 22, something like that. Okay? So what we're concerned about are homologous chromosomes, and if we actually look like this, homologs, right? One from mom, one from pop, but there's some crossing over going on, so there's actually been more exchange. And then like a chromosome from 2 and a chromosome from 3, they're called heterologs, different. Okay, a karyotype displays the different chromosomes. So this is a homologous, homologous. Now, and these are 2, okay? You can also get a karyotype just with a single for each gamete. So instead of having 2 pairs, one from mom and one from pop, a gamete is haploid. It'll either be from mom or it'll be from pop. Okay? And then when the gametes meet, they form a zygote and we're suddenly back in the diploid phase. Okay? Okay. So you will not see the karyotype until it actually condenses during early mitosis, prophase. Even when it's being duplicated in the cell cycle during the S phase, it's being duplicated, but it's pretty much still all strung out, and you really can't see it inside the cell. It's only when duplication is happening, in condensation, where you actually start seeing the different individual chromosomes. Okay, and diploid cells, and then haploid cells. So this is a diploid karyotype, and this is a haploid karyotype, whether it's from a mother or a father. If it's the mother, it's in the eggs. Those are the gametes for the female, and then sperm are for the male side. Okay, meiosis occurs in the germline. Now, meiosis is where the gametes are actually formed. Okay? It's actually, in some ways, very much like mitosis. But there's 2 different phases for meiosis. There's meiosis 1 and meiosis 2. And instead of forming 2 daughter cells, you actually end up forming 4 individual gametes when you synthesize and then go through meiosis. We'll look at that more in detail, okay? And then, of course, fertilization, restores diploid. In other words, you have chromosomes that are homologous chromosomes. How would you design this? Well, it actually starts like mitosis. You replicate the DNA. And it actually ends like mitosis. You separate the sister chromatids. But in the meantime, so let's take a look at this. Okay, so this is mitosis, right? We go from G1 to S phase, where you're doubling the DNA. Then you go to G2, and then finally to mitosis. Prophase, early prophase, late prophase, metaphase, anaphase, teleophase, and then cytokinesis. Okay? Now, this is mitosis, just like meiosis. Okay, so first of all, you have these parent cell. You have chromosome duplication, right? And this crossing over of homologous chromosomes. What happens is, let's take a look here. You're exchanging well, in fact, you're going, let's see here, do that. Okay. What's going on? Let's take a look at one single chromosome. Okay. In this case, you're getting a crossover and you're getting a little piece of the blue when most of this is actually the red. Okay? And then the other chromosome, you got the blue, but now there's been crossing over from an adjacent chromosome and you have a little piece of the red. Oops. Red. Okay, which homologous. There's the same genes, but they might have different alleles. So this is another way that you get greater diversity. Okay, so they're not having duplicate chromosomes. They've been slightly altered so that there's an actual exchange of genetic material between homologous pairs of chromosomes. Now, this is called prophase 1. The tetrad is formed by synapsis. Okay, so now you've actually got these 4 chromosomes together, right? And during metaphase 1 for meiosis, these 4 homologous chromosomes line up like we do in metaphase, and then they split into 2 daughter cells during meiosis 1. Okay, this is basically anaphase. Now, let's take a look at this. Why does it have red there? What caused that? Crossing over. Okay. So essentially, now remember, you've got a parent, right? And they've got chromosomes from their mama, chromosomes from their papa. That's when crossing over happens. Between their mama's chromosome and their papa's chromosome, they've been crossed over and exchanged genetic material. So eventually you'll have haploid cells. And in that case, instead of just having a copy, let's say from mama, or a copy from Papa, you've got mainly a copy from Mama, but then you've got Grandpapa mixed in there. Okay? So you've got two parents. They have two themselves. They either have one from their mother, that's Grandpa or Grandma, and then one from their Papa, Grandpa. And then crossing over, the grandpas might then have some maternal grandma's alleles on it, okay? So that increases or maintains genetic diversity. Okay. And then, okay, so we've got anaphase, and then we've got the beginning of metaphase 2. Okay? Let's actually take a look at this. Okay. Now, metaphase 2 goes through the same sort of things, but what happens is you end up then with a number of different daughter cells. So let's say from the male line, you'll end up actually with 4 sperm cells. And each one will either have this chromosome, or this chromosome, or this chromosome, or this chromosome. Okay? So you've got actually 4 haploid cells. And those are the gametes. You've got 4 sperm, but each one has a chromosome that's different from the other ones. Okay? Now, and the same thing goes for the eggs with daughter cells. You end up with... Now, the thing is, there's no further chromosome duplication. The sister chromatid separated anaphase 2, okay? And each of these are going to form an egg, except that with the females, actually, there's a bunch of polar bodies that are actually discarded. So I forget exactly how many, but the main thing to realize is that you're going to have 4 haploid cells instead of, like mitosis, 2 diploid daughter cells. We've got actually, there's, I think I've got a video that goes into this, and then diagrams as well, okay? Okay, so starts like mitosis. One special cell division is inserted here, okay? Alignment and separation of homologous chromosomes. And then this is what basically meiosis 2 is like. So it's pretty much like mitosis. You've got meiosis 1, goes through everything, telophase 1, and then finally, right here, this is when you go through meiosis 2, and instead of having daughter cells that are diploid, you have daughter cells that are now haploid, either sperm or eggs. Okay. So meiosis 1 starts like right there, just like mitosis, and then meiosis 2. Okay. Now, so the fertilized egg is called what? What's a fertilized egg called? Zygote. And is it diploid or haploid? Diploid. Okay. Okay, so, oops. Okay, why have sex? Well, I guess I could say because it feels good. I might get in trouble for that, but in fact, asexual reproduction is actually more efficient. In fact, it's twice as efficient in terms of fitness. If you have a cell, let's say you have 10 cells that are asexual, and each round of reproduction, they produce then one cell apiece, so you end up with 10 daughter cells, right? When you have sexual reproduction, let's say you have 5 males and 5 females. That's 10 individuals, but they only produce one individual apiece, so they end up producing 5 individuals. Only half as many offspring as being asexual. In fact, there shouldn't be sexual reproduction, tell you the truth, because it's a twofold cost to undergo sexual reproduction. You can make twice as many offspring by not having sex than having sex. Now, if that's the case, what might be the reason? Because remember, in terms of fitness, sex should not happen. Natural selection should disfavor sex because you could leave twice as many offspring as a sexual organism. Okay? So there has to be some reason for this. Now, it turns out there's a lot of organisms that only undergo asexual reproduction. They don't have sex. They usually, get rid of that. I hear you. Let me just save this for once. Come on, stop that.